Today we are going to discuss about ICH guidelines. ICH guideline means Harmonization for Better Health. Mastering quality, safety, efficacy, and multidisciplinary requirements. Target audience for this training: quality assurance, regulatory affairs, and R&D teams. Training objectives: understand the history, mission, and structure of the ICH official site. Demystify the four core guidelines categories: Q, S, E, and M. Review critical subcategories such as stability, validation, GCP, and risk management. Implement compliant processes within internal pharmaceutical development frameworks. Introduction to ICH. What is ICH? International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use. ICH established in 1990 by regulatory authorities and industry experts from Europe, Japan, and the United States. ICH core mission: global harmonization to reduce redundant testing, accelerate drug approval timelines, and maintain strict standards for safety, efficacy, and high quality. The 5-step harmonization process. Step 1. Expert working group creates and refine the initial technical draft. Step 2. Assembly signs off on the technical draft, opening it for public consultation. Step 3. Regulatory bodies issue the draft for public comment and feedback across regions. Step 4. Finalization of the text based on feedback and formal adoption as an official ICH guideline. Step 5. Regional implementation into national, local regulatory laws and policies. The four pillars of ICH means Q, S, E, M. Now coming to Q. Q means quality guidelines. Focuses on chemical, physical, and pharmaceutical quality assurance. Coming to S means safety guidelines. Uncovers non-clinical in vitro and in vivo safety strategies. That is called toxicology. E means efficacy guidelines. Mandates clinical trial structure, conduct, safety profiling, and reporting. Coming to last one, M. Multidisciplinary guidelines. Cross-cutting topics such as electronic submission, standards, and dictionary terminology. Now, deep dive. QA. QA is subdivided into A to F. For stability testing, it determines shelf life and recommended storage conditions across distinct global climatic zones. Q2. For analytical validation, it gives framework for validating analytical methods such as accuracy, precision, specificity, and detection limits. Coming to Q3, this is also divided into A to E. For impurities, it identifies thresholds for reporting, identifying, and qualifying impurities, residue solvents, and elemental contaminants. Q7. Good manufacturing practice. Comprehensive guideline for manufacturing active pharmaceutical ingredients. Q8. Pharmaceutical development. It defines quality by design and manufacturing space concepts. Q9. Quality risk management. It gives systematic tools like FMEA and FDA to assess, control, and review product risk profiles. Coming to Q10, pharmaceutical quality system. Modern quality model spanning the entire product lifecycle. Coming to Q11 for development and manufacture of drug substance. It bridges the gap between pharmaceutical development and manufacturing to ensure long-term product quality and consistency. Q12 Lifecycle Management. Defining a framework to manage post-approval chemistry, manufacturing, and controls changes efficiently and predictably. Q13 Continuous manufacturing of drug substances and drug products defines continuous manufacturing as a manufacturing approach involving the continuous feeding of input materials into the transformation of in-process material within and the concomitant removal of output materials from a manufacturing process. Coming to Q14, this is a last guideline for Q, Analytical Procedure Development. It defines a harmonized, systematic, science and risk-based framework for the development, optimization, and lifecycle management of analytical processes used in the pharmaceutical industry. Now coming to S series, Safety Guidelines Overview. S1, which is subdivided into A to C for carcinogenicity testing, establishes the need and conditions for long-term rodent carcinogenicity studies. S2 for genotoxicity testing, core battery of tests to identify compound-induced genetic damage. S3 is also again subdivided into A to B, toxicokinetics and pharmacokinetics. It assesses systematic drug exposure in non-clinical toxicity models. S5 for reproductive toxicology evaluates chemical effects on mammalian reproduction and embryo development. Coming to E series guidelines. E means efficacy guidelines and clinical standards. E2 is subdivided into A to F, pharmacovigilance and safety data. Unified protocols for processing and reporting expedited and periodic post-market adverse events. E3 for clinical study reports, exact structural blueprint required for writing clinical trial reports. E6 good clinical practices, GCP, the international ethical and quality standards for designing, conducting and reporting human clinical common technical document. CTD, standardized format for compiling registration applications, module 1 through 5, submitted to global health authorities. Summary and best practices for compliance. Stay updated. Regularly monitor updates via the ICH training library, cross-functional training, ensure R&D, clinical trial teams and manufacturing operators know relevant codes. Risk first embed ICH Q9 risk assessments deeply inside daily standard operating procedures. Global standardization. Prepare files directly in CTD format to enable smooth global market submissions. Coming to question and answers. Open forum for audience, inquiries, discussion on internal implementation, bottlenecks, contact info for internal regulatory support. Thank you. Thank you all.